Wolfram Zimmermann, Universitätsmedizin Göttingen. Tissue-engineered heart repair.
Good afternoon. What you see here is a typical presentation of a heart failure patient. This is shortness of breath, very much simplified. This disease seems not so dramatic, but if you look at the numbers, you will appreciate that we have 6.5 million patients in Europe being affected, and 50% of those patients die within 5 years of the diagnosis. The problem is that we do not have any therapeutic interventions to date that really address the underlying problem, that is muscle loss. This is why we and other groups have been developing strategies to remuscularize the heart. And the remuscularization in our case is done via tissue patches, which are considered to replace scar tissue and then eventually integrate into the heart and support the heart either as a patch or as a pouch. On this slide, you can see the basic principle of tissue engineering of heart muscle patches and pouches. We basically take cardiomyocytes, we take fibroblasts, and we take collagen. These three components are mixed together, and eventually you can use these components to generate pouches and patches of pretty much any size for applications in animal models, but also eventually clinically. We have been studying this for 20 years. We have developed concepts, first of all, in the rodent model. We can design these tissues under GMP-compatible conditions. We can scale them. We can basically freeze the cells. We can produce them on demand. We have tested them in pig models. We have demonstrated feasibility in pig models and are presently developing non-human primate models, rhesus macaque, to simulate a clinical study design directly in this monkey model as a surrogate phase 1 clinical trial type of approach, and then move this into a clinical application. Obviously, we are interacting closely with regulatory authorities, with the EMA, with the FDA, to basically bring these approaches into clinical application, and we have founded a spin-off company which is basically developing the GMP processes for clinical application. We want to individualize those approaches. We can develop basically tissues based on models. We are using MRI imaging to model the defect of the heart. We are creating hearts or templates of the hearts by 3D printing, and eventually define the function and structure of the defects and then build the respective tissues according to this. So eventually, the plan is to have, if you have a patient that needs a tissue patch that needs muscle, we only need the information on their HLA phenotype. We basically need the MRI images, and based on that, we can create tissues which we then generate within 2 to 4 weeks and basically can shuttle them back into clinical application. Thank you very much for your attention.